Okay, so let's talk about Chapter 4, cells. And so first a history lesson, because at one time, people were not aware of this microscopic world that we are now and didn't really know what things were made of. And so it really wasn't until the mid-1800s when a couple people started to look at this microscopic world, and one of the first was this person, and he was a Dutchman, and he built sort of the first microscopes. His name, Antony van Leeuwenhoek. He has a heck of a name. Leeuwenhoek. It's an O-E-K. Like I said, he was a Dutchman, and he built, and this is actually a simple type of microscope, which he started by using drops of water as lenses. And so, like I said, he was the first to really start to see this microscopic world. He would look in pond water and in his spit and various types of body fluids and such, and he named what he was seeing animalcules, because he would see things moving around and he thought they were tiny little animals in there, which some of them may have been tiny animals, but a lot of them were probably this group we know as the Protista now. Alright, so von Leeuwenhoek, and then an Englishman named Robert Hooke. He came along and basically built a better microscope and was able to magnify things more and see greater details. And so he said that he basically took a look at cork, which are dead plant cells that come from the bark of plants. And so based on what he saw, he coined the term, or the phrase, cells, because they look like the cells in a monastery, which are the rooms in a monastery, which you can also think of the rooms in a jail, the cells. They were nice, orderly, about the same size and everything. Okay, so Robert Hooke came up with the term cells, and here are some animal cells. We'll look at some of those later. Now we get to these guys, Schleiden, Schwann, and Virchow. For some reason I always like to say Schleiden's name first. I don't know why that is. And so von Leeuwenhoek and Hooke were looking at this microscopic world and saying, oh, there are these tiny little things, and we call them cells. And so these guys came along and said, well, are all things made of cells? Are all plants or are animals made of cells, for that matter? And so they did research on different types of organisms, and indeed they confirmed, in fact Schwann confirmed, that animals are made of cells, and Schleiden that all plants are made of cells. Now Virchow's contribution here is that basically in order to get new cells you have to have old cells. So new cells come from, we would say, pre-existing cells. They don't just pop out of nowhere. And so all of this gave rise to the cell theory. And what does it say? It says all organisms are made of cells. Okay, whether you're a single-celled organism, a multi-celled organism, they're all made of cells. 2, cells are the basic units of structure and function in living things. That is, cells are in there doing all the work and providing the structure for living things. So the cell's where it's at, you might say. And the third part of the cell theory is Virchow's contribution, again, that new cells come from pre-existing cells. Okay. Now, that begs the question, okay, where did the first cell come from then if you have to have pre-existing cells to make them? Well, that is a question you can think about for now. We're going to visit that a little later, but keep that in mind. Okay, cells generally are pretty small things, okay, from bacteria that are quite small, bacteria in the range of basically 1 to 10 micrometers. Remember that a micrometer is 1/1000th of a millimeter. And then eukaryotic cells, so those are prokaryotic. Eukaryotic cells typically in the range of 10 to 100 microns in size. And those, of course, are the things we need a light microscope to see. The book discusses in this section, this chapter, talks about light microscopes and electron microscopes. We're going to only use light microscopes in this class, but we just want to keep in mind, we don't have to know the details of an electron microscope, but they're basically used to see really small stuff. So while your light microscope can make out things like chloroplasts, when it comes to things like viruses and certain types of molecules and even individual atoms, you need an electron microscope to see those really tiny things. Okay, why are cells, why do they tend to be so small? Well, it's sort of a biomechanical necessity, because if you think about it, an analogy, now you're in a very large building and you're at the center of that building and you just start walking towards the edge of the building, and it'll take a while to get there. Think you're in the new convention center downtown. It's a gigantic building. If you're in the middle of that, to walk to the edge to get out is going to take you a little while. Whereas if you're just inside my classroom here and you're in the center of the classroom, you can walk to the edge and out the door in a matter of seconds. Whereas it might take you a matter of minutes to do it in a very large building. So it is the same with cells. They tend to be small because it makes it easier to get things in and out. There's less distance to travel, and also as something, when you have things of the same shape but of different sizes, and think of these as three-dimensional balls, as you're going up in size, of course, the volume increases, right? The surface area increases. This has more surface area, and this one here has even more surface area. But which of those increases faster? Well, surface area is increasing to the square root because it's a two-dimensional thing, whereas volume is increasing to the cube root because it's a three-dimensional. There's an x, y, and a z axis in volume. So what happens is, as an object gets bigger, of course the surface area increases, but the volume increases at a faster rate, such that in larger objects like this guy, your surface area to volume ratio is less than your surface area to volume ratio in your small object. So smaller objects have more surface area relative to the volume, and it makes it easier to get things in and out of that cell. Shape is related to function. In skin cells, for example, they tend to be flattened discs that form a multi-cell layer to keep things out and in your cell or in your body. So they serve this protective function. Cells that line your digestive tract tend to be in a single layer, and they're used to taking things in and releasing materials. So that single layer helps facilitate that, and particularly absorbing nutrients and such. Your nervous cells, long and thin, to help transmit signals around the body. Your red blood cells having these nice rounded edges, making it easy for them to flow around your bloodstream. White blood cells having sort of an amoeba-like shape in that they can change their shape and slide in and out of narrow spaces. We'll see how your red cells are pretty much stuck inside the blood vessels, but the white blood cells can squeeze out to go into surrounding tissue to help fight off pathogens. Your platelets, which are sort of cell-like structures that in your blood that have to do with clotting, and so they don't have a nice round shape. They sort of have edges and such that they can block off the blood flow in a cut. Now, microscopes, of course, allow us to see things. The light microscope, see small things. The light microscope we use, again, its advantage is it allows us to see things like cells that are relatively small. That thing we're looking at can be alive while we're looking at it. Disadvantage is it has limits in how small something is that you can see, and also the sort of the level of detail is limited. Now with electron microscopes, and light microscopes are relatively cheap. Electron microscopes allow us to see really small stuff with significant detail. So you can see very small things and significant detail. Disadvantages: expensive. They can tend to be a little large and bulky, although the newer ones are getting smaller. And the process of preparing the sample to see it basically kills the specimen, so you can't see it in its live state. Okay, there's section 1.